Okay, so hello everyone. So I'm not going to make this opening speech. It's going to be later after the evolutionary session in the stream to lecture. It's my great pleasure to start this 17th edition of Evolutionary Behaviour conference, and we will directly start with the evolutionary session. I am honored to present our first speaker, so Alexandre is here. He is a senior researcher in evolutionary ecology at the Leibniz, I don't know how to pronounce this name, Institute for Zoo and Wildlife Research, and he is the head of the data zoo game team and does teaching at the Freie University in Potsdam. Alexandre is from here. He actually did his PhD in Montpellier, but in the other evolution lab at the Institute for Evolutionary Science in Montpellier. and he followed with a postdoc at the University of Sheffield, and in both his PhD and his postdocs, he worked on humans. He then was a Marie Curie Fellow at the really don't know how to pronounce this, I'm sorry, in Berlin, and he's a quantitative wildlife researcher at Leibniz since 2012. His research focuses on the evolution of life history traits, particularly the link between individual and population level processes. And he's interested in mammals like spotted hyenas and bats, but also in birds with parrots and birds of prey. He works with data from natural populations to explore patterns like individual variation in traits, mate choice, and population demography. And he is also committed with open science by developing and contributing to R packages to help other researchers to do statistical analysis. And today, so in his talk, Counterintuitive Patterns in Ecology and Evolution, or why we should not forget that a population is made of individuals, he will show how population level patterns can mislead us in our conclusions in evolution. Alex.
you very much, Lisa. Not sure how to do that. So yeah, thanks. It's really a pleasure to be here today and to start this first session of this very exciting conference. Before I start, I wanted to make a little confession. So the little confession is that I was, of course, delighted to be invited, but a little bit terrified. But also I had a problem. And the problem is that since I finished my PhD more than 15 years ago here in Montpellier, I've been jumping from research project to research project, from species to species, and even from discipline to discipline, which doesn't make it particularly easy to build a long, inspiring, coherent narrative for a plenary talk like this one. But I'm gonna try. And I think I'm gonna mostly build on an insight I had no later than 2 years ago. This is very sensitive. So I'll leave it there. So yeah, 2 years ago, it was November. I was not sleeping and I was wondering, what the hell am I doing in research? And I had a little like eureka moment. It crystallized in my mind. And I realized that all the research that interests me the most, in fact, boils down to a single question. So this question, how does a particular pattern observed at the level of a population come about? And I think a lot of you and a lot of people out there are working on population level patterns, but they don't necessarily do it explicitly. I certainly wasn't doing that explicitly for many, many years. And I think it's a little bit of a lost opportunity. There is one exception, is people working on collective behaviors, like people working on enumeration of starlings. Those people, they really try to connect tightly the individual level on the collective level. So for instance, here you see a enumeration, each line is so exact photographies. Each line represents what a single bird does. And these people, they study the behavior of every single bird, and they try to understand how these percolate to this group level pattern. So I'm gonna try to take this as an inspiration to revisit some of the research projects I've been doing for some time. I've chosen 3 of them, I don't have much time, but I'm gonna try to be more explicit about this idea of connecting the individual on the collective level. And I hope that by the end of the talk, I will have convinced you that it's a worthwhile things to do. So let's start with the first population level pattern, which is the choosiness. Choosiness is the intensity of mate choice. So mate choice, a lot of people know that has really strongly shaped evolution. It shaped evolution because it influenced the selection on trait through sexual selection as well as cultural selection. What is less clear, though, is why mate choice, the intensity of mate choice, the thing I told you we call choosiness, why does it vary so much? So it's very difficult to measure that in a uniform way, but there are various proxies out there, and this is one. So for instance, how often do you mate with the first individual you encounter? And you can see there are big differences. You have differences between species, you have differences between populations, even within population, you have differences between species, between sexes, sorry, you have differences at all scales. And the question is, why do we have such variation? So we wanted to understand that, and we wanted to try to find what are the minimal conditions that generate part of this variation, at least. So we tried to make a very, very simple model. So we were, okay, let's first consider that mate choice is just one value. This value P of choosiness, it's a threshold. If you meet a potential partner that has a quality higher than this threshold, you say yes. If you meet an individual that a quality lower than this threshold, you say no. So it's a threshold function, very simple. Okay, you say no, you say yes. So here, this is the minimal quality you're willing to accept in a mate. There are all kinds of qualities that have been analyzed out there. There are things called indirect benefits because the qualities don't benefit you directly in terms of fitness, but it does benefit your offspring. And there are also like direct benefits. So this is something that directly brings benefit to you. So the partner brings something and you benefit from it directly. So example as for instance, if we consider female choice, an example could be that male provide territories, they can provide food, for instance, as nuptial gift in many insects. They can provide protection of the children or protection of the female itself. And sometimes male can even contribute to parental care. And if I would take male mate choice, we could say more or less the same thing. So we were, okay, we're gonna consider very simple benefits, the ones that are direct, and we're gonna consider that they directly influence reproduction. In this context, how can we explain variation in choosiness? One obvious idea is to say, well, there's different costs between species or sexes or population for the evolution of structure that allow to do the choice. But we were not really comfortable with that because we thought that this on its own cannot explain a lot of the huge variation we observe, because there's often like very similar species in very similar environments and yet very different intensity of mate choice. So we said, let's scrap that. Let's make it super simple. Let's consider that choosiness bear no physiological cost. Okay. So now, if you think a little bit classically, you would be like, okay, in this context, there is benefits, there is no physiological cost. So what should happen? Should be selected for, yeah? No. Should be. And usually it's what people think, and that's why a lot of people didn't do that kind of model, because then if it's selected for, everybody's got high choosiness, you've explained nothing, doesn't work. But what I will show you is that your intuition is wrong. At least maybe you didn't have this intuition, but those who thought, one, the intuition is wrong. To understand that, you need to stop thinking about an individual in isolation, and you need to think about an individual in a population. When you do that, you realize that when you're more choosy, there is an unescapable cost that emerges. And it's the fact that it will, in all systems, it will reduce the mating rate. It does it for two reasons. So first, if you want only high quality partner, it's a subset of all the potential partners. Okay, so there are less of them. And also, if you have several individuals who prefer high quality partner, they're gonna compete between each other for the access to this individual. And they compete because the partner you want, if the other people want them, they may be already mating and therefore they cannot mate at the same time. There may be gametic depletion. They may be doing parental care and therefore they cannot mate at that moment as well. So there's all kind of reasons that can lead to that. So how do we then, once we recognize that, how do we understand the evolution of choosiness? Well, we can adopt the formalism of mutant and resident to look at evolution. So it's something called, well, it doesn't matter what it's called. But basically, you look at the payoff of a mutant strategy, so very, very rare strategy, when all the other individuals are doing something else, and we call them residents. So for instance, in this case, the mutants, the lower threshold. So it means that for all potential mates that have a quality between this value and this value, there will be no competition. The mutant will have access to them, and those individuals, they will not be busy mating because no one else wants them. So once we've put this in place, we have to assume a particular life cycle. And we also did something very simple. We say individuals, they can be available, they can be unavailable, or they can be dead. Once you're dead, you remain dead with a probability of 1. That's the simple case. No zombie here. But all the arrows are probability of transition. And the interesting one is this one, when you go from available to unavailable. So SEML. So SEL are 3 rates. So S is the probability you survive to the next time step. E is the probability you encounter an individual of the opposite sex. And L is the probability that if you've mated, you become unavailable, or if you are unavailable, that you stay unavailable. Okay, we made it simple. We could have split that into, but we didn't. The tricky thing is N here. N is the mating probability. So yeah, so you need to survive, encounter somebody, but then you need to mate with it. And this is a function of all these rates. It's a function of the choosiness of the mutant, of the resident, and it's also a function of the distribution of quality in the population. But what we found mathematically is that there is always one value of choosiness that is an evolutionary stable strategy. So it means you're gonna evolve, given the assumption of the model, to a particular value of choosiness, and you see it's not necessarily an extreme value. This is because there is a trade-off between the benefits per mating and the fact that the number of mating decreases when choosiness increases. Okay, there is this emerging, unescapable cost. And then when you look at, okay, what happened when the environment changes, when the genes change, and this is reflected by changing the free rate I mentioned before. So for instance, if the encounter rate is lower, you meet less individual. This reduces the number of mating, of course, but the shape changes, and that leads to select a lower value of choosiness at equilibrium. So you see that with modification in survival, in latency, in encounter rate, we can already create variation in choosiness. We in fact show that we can predict this variation qualitatively based on how it changes what we call the RST, relative searching time. So it's the proportion of your lifetime you spend searching for mate. If something increases this proportion, you spend more time searching, that will lead the choosiness to evolve to a lower value. If something reduces the time you spend searching for mate, that will lead the choosiness to evolve toward a higher value. So this was the first puzzle that we cannot solve. So everything is possible, in fact. And the reason, the fact that we have this counterintuitive pattern is the fact that we assume that mates are not unlimited. Many models in sexual selection, they actually have this polygyny, this infinite polygyny assumption, the idea that preferred individuals, they can mate with everybody. We think that never happens. And that leads to frequency-dependent competition among choosers, which leads to this trade-off. And therefore, that limits the evolution of choosiness and shape what choosiness is maximal. What I like in this thing is that it also shows that classically in sexual selection, people, they compare intersexual selection to intra-sexual selection. I don't know if you've heard those terms, but by inter, often the typical example is female mate choice, and by intrasexual selection, the typical example is competition between males. But here I show that if there is choice, so in the intersexual component, you will automatically have intrasexual selection among the choosers because of this competition. So the two are entangled. Okay. Now I want to take you to a completely different place. Hopefully you're going to see some common point as I go. So I want to bring you here. So this is in the middle of the Ngorongoro crater in Tanzania. If you have the opportunity to go there, you should. It's more wonderful. But don't spread the word. So this is a place where some colleagues of mine, and there are some in the room. There is Eve somewhere there, and there's Marta. I don't know where Marta is actually. Yeah, Marta there. Study spotted hyenas. The population has been monitored under the supervision of Oliver Hunter since 1996. So we have more than 29 years of data and very high resolution data. And there's so many fascinating things I could tell you about hyena. But instead, I'm going to choose a relatively boring population level pattern, which is this one. It just shows how the number of individuals has changed over time. And as you can see, it has changed a lot. From 210 individuals, for instance, we reached 550 individuals in 2011, and 620 individuals in 2017. As of January of this year, there were around 477. So there's been a lot of changes, and I was particularly interested in this period of growth here. So when you see a growth in a population, how do you interpret that from a demographic point of view? What do you think? Do you think that survival and or reproduction must have increased? Or do you think they could have decreased? Sounds weird, doesn't it? Or do you think they could have stayed about the same? Who thinks 1? Who thinks 2? 3? Who doesn't think? Okay. Yeah, so if you think 1, I think you are a rational human being, but I will tell you that it's wrong, okay? So why is it wrong? Well, first, I'm showing you it is wrong. So here, you see, we have the monthly probability of death. It did not go down during the period of growth. And we have a lot of data. Here we show the monthly reproductive output, so the monthly reproductive probability. It did not go up. if anything, it went down. Okay, so you have the population going up, survival doesn't change, and reproduction, if anything, goes down. Okay, so what's going on there? Well, again, if you stop thinking about individual in isolation, but you think about individual in a population, this starts to make sense. If you think in terms of what people call population dynamics. Because in fact, as long as there are more babies than individuals dying, the population will grow, irrespective of what happened to survival and to mortality. Okay, so often we say, well, it's just that the population has not reached its carrying capacity. Okay, so the population gonna join the carrying capacity, and different things can happen while this happens. So I'm gonna try to kind of untangle all of this to try to understand a bit better what's going on. So first, what is population abundance, really? It's the number of individuals alive at a given time. Okay, we can also call it population size, census size, whatever. And it reflects, and this is the important thing, the effect of past environmental conditions. It doesn't reflect what's happening right now. It reflects what's been happening during the recent past, but also the past a long time ago. All of this lead to the number of individuals we have today. On the other hand, the carrying capacity, the best way for me to define it is like this. It's not something biologically like real. It's an abstraction. I think it's a useful abstraction, but it is an abstraction. It's a human concept. And what would be the expected number of individuals that the population would eventually stabilize around if conditions would stop changing? Okay, and usually we use that to reflect the effect of environmental conditions experienced during a certain time period. And people, I don't know why, but most of the time period they consider is the whole study period. So they see carrying capacity as a fixed property. And the typical thing you will find in the textbook is this. If the population starts low, but not too low, or let's forget about Ali effect and things like this, the population will go up until it stabilizes around the carrying capacity. Yeah, perhaps. But you can imagine that the carrying capacity changes. And then things start getting interesting, especially in this case. Because here, what do we have? We have a phase where the population goes up, while in fact, the carrying capacity goes down. So that means that the environment is deteriorating, the vital rates, the survival, the reproduction go down, and yet the population is growing. This is a nightmare for conservationists, because it means you do things on the ground, you count your individuals, the population grow, you're like, yes, I'm doing good work. And in fact, maybe you're just destroying the potential for your population. Because if this doesn't start to fall down, like then the population, once it reaches carrying capacity, after it will dwindle, will collapse, okay? So we wanted to estimate how carrying capacity changes with time, because the thing I showed you is like a video game, like it's monotonous, like it goes up or it goes down, but the reality is much more complex. So we combine a statistical model fitted on the real data to an agent-based model. I really cannot go in detail. It would take us like months. For us, it took us many years. But the idea is that on the real data, when you fit this model, you have a year random effect, which captures the influence of the environment. So then when you use this model inside your individual-based simulation to predict what's going on, you could fix the realization of the random effect. So it essentially simulates that the environment stops changing. And then you could wait until your population will stabilize around a value. And this value is the carrying capacity. And then you change the year and you do this again and again and again. And of course, you do this many times for each year. And that leads to a plot like this. So in red, we have the change in carrying capacity over time. And in blue, we have the change in population abundance. So what do we see here? We see that right from the beginning, the carrying capacity was much higher than the population abundance, which means that in terms of vital rates, the individuals were doing fine. They were reproducing a lot. They were surviving well. So the environment had already recovered, but the population, for the population to recover, it took more than a decade. Okay. So here, what we're observing is that because the population is not at equilibrium, there is a long demographic line. And it's not particularly surprising for species that are long-lived and don't reproduce very much every year, like the spotted hyena. So I showed you that, contrary to our intuition, number one could have been possible, but everything else could also have been possible. And in this particular population of spotted hyena, the reality is closer to this. So survival didn't really change, but reproduction decreased a little bit. Okay. And I mentioned that before. So far, so good? Okay. So we can move on to yet again something different and yet quite the same. Twinning in humans. So I don't know how much you know about twinning in humans, but there are two main kinds of twins. We have what we call monozygotic twins, that is identical twins. And we have dizygotic twins or non-identical twins. From a biological point of view, those two types of twins are very, very different. I mean, besides the fact that some are like, you know, almost clones and some are more like brothers and sisters, there are huge differences. So first, monozygotic twins, they only occur at a very low frequency, 3 to 5 per 1,000 births, all the time, everywhere. The frequency doesn't change with time, it doesn't change with space. There is no heritability on this trait. In fact, they are so rare that in movies, they usually take the same actor to make you believe they are twins. That's the case in Sinners, as you may know. The other type of twins is more interesting from an evolutionary point of view because the frequency can become quite high, several percent of all birth. The frequency varies in time and space, and it's highly heritable, and it has a huge cost in terms of survival for both the mother and the children. Even in modern hospital, it has a huge, also financial cost. So why? Why can't we have such frequent dizygotic twinning despite all this cost? So at the heart of the hypothesis to try to explain twinning, there is this relationship. The relationship between the lifetime twinning probability, so whether you are a mother that has been a twinner or not, whether you ever produce twin or not, and the total number of birth. And birth counts for one, whether it's a singleton you produce or twins. So people based on that have argued, well, the female producing twins are female with, I don't know, very high quality. So they can make a lot of birth, and with each birth, they can even make 2 babies rather than 1. But if you think of that, you're like, well, then, like, why are the frequency of twinning as they are? And this, I computed this using pre-industrial Europe, but it's been replicated in many, many populations. It's a very robust pattern. Which brings to the last puzzle. If that's the case, what would you expect twinning to be selected? How would you expect twinning to be selected? Selected for? So it would increase. You know, it increases lifetime reproductive success. What do you expect the trait to be? Increasing in frequency, yeah? I will show you that's not the case again. So why not? Again, you need to stop thinking about individual in isolation, and you need to start thinking about individual in a population. So here we have two possibilities, and both could be true. But the first is that high fertility female have higher twinning propensity. That's what everybody thought until we started to work on that. But the second is a bit more subtle, is that high fertility female may just have a higher exposure to the risk of twinning. So it's a bit like if you play the lottery many times, even if you win winning ticket, even if your ticket has the same probability of winning, the more you play, the more you can win. Okay? So to really understand what's happening at the level of twinning, we need to control for this variation in exposure to the risk of twinning that is mostly driven by environmental stochasticity. And when we do this, when we control for exposure to risk by analyzing not the probability of producing twin during your life, but the probability of producing twin at every, like for a given birth, you see that the relationship flip signs. This is cool. In statistics, I do a lot of statistics, super cool. This is called a Simpson's paradox. It's the most extreme form of ecological fallacy. And usually this is demonstrated when you aggregate individuals. Here we find that it happens when you aggregate birth within an individual. So, but beside this kind of geeky excitement, this shows something a little weird. It shows that mothers with twins have higher fertility, but at the same time, mothers with higher fertility have lower twinning probability. So that sounds like a little bit like a brain fuck. Let me help you there. This is completely equivalent to the situation, which I think is much easier to understand. People who drive more have more accidents, and people who drive more are less accident prone. Both can be true. Taxi drivers, for instance, there are a lot of data on that. By the way, this is a taxi in Berlin that got squashed between a tram and the pillar, cut in two. So taxi drivers have a lot of accidents, but they also drive day and night, every day, most of the year. And in fact, if you look per kilometer, the accident rate is much lower than the average citizen. So if you go to the airport, better, if you want to avoid accident, better you go with a taxi driver, even if the taxi driver had accident last year, rather than a grandmother, for instance, that may not have had accident, but may not drive very much. So you understand that we need to control for the exposure to the risk. So the positive correlation that I showed you first between twinning and fertility, it emerges from the aggregation of these birth events, and it doesn't require physiological explanation. What does require an physiological explanation is this negative relationship here. We used something a bit similar, but very different from what I presented before, but it's still a combination of statistical models and simulation. It took us, again, years to get there. But at the end, we were able to identify the mechanism that explain most of this negative slope. It's very simple. So especially this one. This is just the fact that once you have twins, the probability you keep reproducing goes down. You know, after having twins, you stop. It's pre-industrial data. Maybe you stop because you're dead. But there is another thing that's a bit more subtle, and here on the slide I've cut a little bit corners, but basically we know that twinning changes a lot with females' age. This has been demonstrated all over the planet. It peaks around, so it depends on the population, but between 35 and 40. And because of this, depending on when you produce in your life, it will shape this relationship between twinning and fertility. So if you reproduce late, you will have quite high rates of twinning, and you cannot accumulate that many births. Remember, the maximum is 18 births in pre-industrial Finland, it was. So you can't do that if you start late, and that shapes the relationship. So this kind of environmental heterogeneity between individuals when they give birth and when they start and things like this shapes all of this relationship. So, but what about selection? So if you kept track, I'm not expecting you to because it goes a bit fast and there's a lot here, but bear with me. We've shown that when you have higher twinning propensity, it leads to fewer births once you control for the variation in exposure to the risk. At the same time, you have fewer births, but because it's twinning propensity, there's more chance that a given birth will give you 2 children rather than 1. So in terms of selection, you can still win. You could do less, but double, or you could do, or maybe you just do less overall. So we actually simulated a very small increase or decrease in the twinning propensity, and we look at the emergent property about what's going on for selection. And we show that depending on the exact parameter value we take from the different population we work on, we could have situation where it's selected for, situation where it's selected against, and in between situation where there's hardly any selection. So it's very easy to actually create variation in the direction of selection and intensity of selection because of this negative relationship. So that kind of solved, I think, that largely solved the problem we were trying to explain. So yes, everything is possible. On this time, it's because the variation in exposure to risk introduces some environmental stochasticity that blurs the real pattern. In other words, technically, people would say, oh, this is the evidence of a strong gene by environment interaction. So now it's time for me to bring these different threads together. And what did I show in terms of counterintuitive pattern? This is 1 minute or 10 minutes? I showed you that the evolution of choosiness under direct benefits is self-limiting. I also showed you that the population can grow while its environment is deteriorating. And I showed you that twinning is more frequent in mothers with high fertility, despite reducing fertility. Okay, that's kind of counterintuitive. At least it was for me. The causes for these different patterns are those one. First, I showed you there is an unescapable cost that emerges from the competition among choosers. The second, I showed you that the population abundance emerges from the combination of past vital rates. On the third, I didn't show that very directly, but believe me on that, individual variation emerges from difference in exposure to the risk of twinning. So you see, I've highlighted emerge each time, like emerges, because I think this is really the reason why things are counterintuitive. I told you that the question I was working on is this one, but in fact, the larger question is really this one. How do emergent properties come about? Emergent properties are usually defined that way. Properties that are not evident in the individual component of the system, but show up when combining these components. And here, I love it that the author, they added, so they might also be called surprises. So why do I like that? I like that because I think this is why I like this research. I like little puzzle. I like when things are counterintuitive. Like there are people who like to demonstrate the obvious. I'm so glad it's not me. We need them in science, but I'm so glad it's not me. It doesn't sustain my interest. The conclusion of looking at emergent structures is that I told you, you need to kind of stop thinking about individual in isolation and really embrace the fact that it's individual in a population. What does it mean in practice? It means two things. So first, try not to make assumptions if you're doing theoretical modeling that kill emergence in the egg. So for instance, in sexual selection, infinite polygyny, just like it stopped the emergence altogether. If you are analyzing data, don't compare aggregated groups. Don't compare aggregates. Don't compare means between groups without having. We won't do the analysis at the level of observation. Okay, this is a deadly recipe. It leads to you could search for ecological fallacy or Simpson paradox. It is a big problem in statistics. So always try to do the analysis at the level at which you have your data. Now, if we step back a little and try to understand, okay, but what were the mechanism and what do they share in common? The first, it was frequency dependence. And I told you that because of frequency dependence, fitness become a function of the genetic environment. So not just your genes, but genes of the others. Remember, mutant resident. The second, the mechanism behind the counterintuitive pattern was this non-equilibrium dynamic. And there, fitness become a function of the temporal change in the environment. Okay? And the third is that we had strong gene bioenvironment interaction and it made fitness dependent on the stochasticity in the environment. So each time you see, we have the environment in the very wide sense here, including even the genes of others. And I think that explains things. I think that the counterintuitive pattern, they emerge as a result of the integration of all these genes and the environment together. And if you look at the literature on emergence topic that physicists work on, they always tell you that emergence happens when you have nonlinear effects that are summed together. And here I didn't show it explicitly, but I know that in each of the cases I've presented, there is very strong nonlinear relationship. So I think that's the reason. And the conclusion is, therefore, a population is made of individuals with an S. I think, of course, everybody knows that a population is made of individuals, but there is knowing and there is really like integrating this information. And I hope that the talk helps a little bit about that. So these individuals, they are shaped by their own gene and environment, but also by those of others. So since a lot of you probably work on population level patterns, one kind of another, I think you should try to embrace the complexity and embrace emergence in all its glory. I think you will learn much more about your system if you do that. It takes some effort, but I think it's extremely rewarding. On here, I just wanted to illustrate, I wanted to show that there are so many interesting population level patterns. And I focus at the interplay between individual and population, but you can also focus at different level of biological organization. There is, you know, every field is kind of the study of the emergent property of another field. People tend to say that it's not always true, but there is some truth in that statement. So just in hyena, for instance, I've been studying recently like mating patterns. This is a population level pattern. Who mates with whom is really influenced by the preferences of everybody in the population, the availability of everybody, the demographic of the population, where individuals are, and so forth. With Eve, we've been working, we will continue to work much more on the emergence of social hierarchy. So what shaped this agonistic interaction between individuals, on why in spotted hyena we have, for instance, an individual in each clan dominating all the others. On Marta today, will also present the comparison of reproductive inequality in male and female in spotted hyena. And she will show you that this is, again, an emergent property. But I'm not going to say anything more about this because that's Marta's goal. So with this, I'd like to thank the people who came in my group that work on this question I mentioned. So Loïc, Liam, Conan, Leonie, and Elisa, as well as the more senior people with whom I've been interacted a lot on these various topics. And I would like to leave you with the references of the paper I mentioned, in case you're interested. And I'm very happy to take questions either right now or if, because I think we're on the record, if you're a bit shy or something, anytime during the week, I will stay with you the entire week. And I'm done, yes. Ah, on m'entendait pas bien sur YouTube? Bon, bah, tant mieux. Okay, so priority to young researcher for questions. Don't be shy. Okay, we have a question here in the center.
Okay, thanks a lot, Alexandre, for this very interesting talk. Just to know, it's a special mic, so I can just. It is very smooth. Don't be shy.
Oh, hello. Yeah, thank you for the talk. I just had a tiny question on the risk of twinning, because I was thinking with the analogy of driving mistake, but I feel like twinning is.
Sorry, could you repeat the technology?
So there was an analogy of taxi drivers getting into accidents, and taxi drivers getting into accident feel like maybe there was a judgmental mistake, but what is the. But twinning doesn't sound like a judgmental risk. So what is the actual risk of twinning? Do the mothers tend to die?
Okay.
That's twins, or?
There's an audio problem here. It's echoing somewhere. I think you've been misled by the word risk. I compared twinning to, yes, the risk on traffic accident. I also compared twinning to lottery. Can we do something about the sound? I just stop this. But basically, if you have individual that reproduces a lot, that have a lot of baby, irrespective of the reason why they do so, it really increases the chance that at least one, they will have twin during their life, even if their probability of having twin is actually lower than some individuals that don't reproduce as often. Okay, what do you prefer? I give you 100. Sorry. What do you prefer? I give you like 100 lottery tickets that have a chance of 1 in a million to win. And I give you 100 of them. Or I give you 5 of them that have a chance of one chance over 500,000 winning. You take the former. Okay? You take the lower winning probability, but because you play more time, you're more likely to win. And that's what shaped this positive relationship. I'm not sure you. It's difficult.
I get it. I was just wondering what was actual risk of win because
No,
it sounds like a benefit.
The risk, the cost, why it's costly to produce twins.
Yeah, if it's either.
Well, first, you need to be able to afford it. The quantity of milk you need to sustain, for instance, to breastfeed the twins is way higher than to sustain a singleton. This is already a problem when there is food restriction in the populations. So historical time that was a problem. Another problem is during delivery. A lot of complication happens during delivery because you have just more that needs to come out one way or another. So the risk of mortality of twins compared to singleton is always higher. And for mothers, the risk of dying is in general 5 to 10 times higher when they produce twins than when they produce singletons. On average, at hospital, it costs in Netherlands 11,000 euros more today when the female produces twins. Because also they're trying to anticipate the problem, and therefore they put much more medical staff, they are more likely to do C-section and all these sorts of things. So this is cost, this is not risk. Well, in my phrasing.
I was always terrible at this. Yes, and my question is about the scheme you had from available to unavailable in sexual selection part. You had latency also involved.
Yeah.
I was wondering, is this the latency to mate, or it's also, for example, latency to give a nuptial gift, or latency to sing, the latency of the courtship, kind of?
So another wording that people are using is often time in, time out. You know, like, are you in the mating pool or are you out of the mating pool? Are you out of the mating pool because of previous reproduction? So the latency here means you get out of the mating pool for some time. I mean, here it's a probability at each time steps, so it's not the same duration for every individual if you want, but it's the same. It's the same idea. What can make you unavailable? What can put you in this kind of time outside the mating pool? It could be so many. For instance, it could be mate guarding. You know, this kind of like a frog, a male frog, and you stay behind the female for hours a day. It could be that.
Okay.
It could be, as I say, gametic depletion. You no longer have enough sperm. Okay. It could be that you're looking after the children so much that you don't have enough energy to reproduce again, like lactobacillus in humans or females. So I think if you really look at natural diversity, you will find plenty of examples that lead to this latency, depending on the type of organism you have.
Okay, thank you.
I don't know if I got it about now. Can you hear me?
Sorry.
Can you hear me?
Yeah, I can. YouTube cannot.
Okay. I'm interested in opportunity. If you go to a graph that is carrying capacity for the highness, number of individuals with carrying capacity, you can open that graph.
I know what you're going to say.
Yeah. Next one. Yes. So I'm thinking about it like predicting point of view. I mean, I know us ecologists like to predict out. I mean, so we can manage this animal can convert. At some point, I'm seeing imaginary lines like on the carrying capacity side, such that if you know trading or trade like market analysis and all of things, if you have those lines where the carrying capacity changes because of the number of individuals and stuff, at some point you can predict that. I think if you use a graph, let's say you have 300 years of data, right? At some point, you know, like if you have a certain number of individuals, and you know, like you have lost too much in life, you cannot predict like the carrying capacity changes in the environment. Probably, I'm just assuming, can use the same statistic from your point of view to when you manage the environment. But if you know the carrying capacity, you know what could be wrong with the environment, for example. Let's say you're looking at it from elephant point of view, elephants affect the environment, the damage and all those things. So at some point, if you know the number of individuals and you can compare historical data, can you think at some point you can say carrying capacity, because now we have so many individuals, male, female, I don't know, you know the potential carrying capacity. Can use that for management purposes.
So I think there's a lot of unpack there, and I think we need to speak some more. But I think one of the things you're touching at is like, how do we estimate carrying capacity? What kind of data do we need for that? Yeah. Unfortunately, because here carrying capacity is an emergent property, we need to be able to model the vital rates quite precisely on the density dependence on these vital rates quite precisely, and that requires a lot of data. I've been thinking a little bit about trying to devise more general model that will allow to do that for many species when you don't have much data. A bit like what they do in population variability analysis. I don't know if you know these things like software like Vortex. You could predict if your population is going to crash or not, even if you know hardly anything about your population, which begs a bit of question like how reliable are the numbers being produced. But we understand this problem. I think it's extremely difficult. I think also here we are lucky because we have had our population go through many different densities, so we can model the density dependence quite well. If that's not the case, if you only have your population at very low density, you don't know what's going to behave this density that's very high. So it is a fundamental obstacle, I think, for the precise understanding of biosphere conservation. And I'm not saying that counting individuals is bad. I think for a lot of people, it would already be good if they would be counting the individuals, because in a lot of conservation projects, they don't even do that. But what I'm showing is that it would be good also to measure like other things to make sure that some data rates are not going down, or that the individual they remain healthy, and that numbers going up alone is not necessarily an indication things are going well. It could be, perhaps even most likely they're going well, but as I showed you, it's not necessarily the case, especially if you have like long-living species. Okay. One last quick question.
I was wondering, for the twin pattern that you found, where lower fertility women have higher rates of having twins, or have a higher chance of having twins, is that just a byproduct of like lower fertility women are more likely to have in vitro fertilization?
No. So the reason I didn't show you the data set and all that. The reason most people interested in evolution of twinning, all work on pre-industrial population is exactly to avoid this problem. Okay. Modern fertility treatment really messes up twinning rates. Should plan to see. So if you work with modern data, it's extremely difficult because often we don't have like high level of detail about the fertility of women. And actually, one of the best efforts really monitor the health of females across the world. It was the DHS program. It's just been shut down by Trump. So after many decades of the program running very smoothly on really getting like super nice level of data to have that discussion. But to avoid this complexity, that's why we go back to pre-industrial records. So it means mostly working with church level records, like the church were quite good at keeping track of how many individuals are born and when they are born and when they die. So there's a lot of studies in Europe, and there's a lot of studies with the Mormons in the US. Okay, thank you very much.
Okay, I'm just testing the mic.
Test, 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 test.
Mais nous, on a un boîtier exprès pour les fous. On va mettre ça.
Bah, tu vois, c'est juste pour la retransmission. Ouais. Un, deux, un, deux. Ok. Ouais, bon, je vais le garder là.
Et je sais pas si tu auras la note qui a des petits posters, genre voilà. Ça pourrait être plus clair. Okay, so thanks again, Alex, for this wonderful talk. So now we are going to listen to Léo Desjeux, a PhD student in the l'ENA in Lyon, and he's going to talk about snails and experiments.
Thank you. Okay, thank you. So first, I would like to thank the organizers for the occasion to talk here. And as she said, my name is Léo Desjeux. I'm working at the l'ENA laboratory in Lyon, which mainly focuses on functional and evolutive ecology. And today I'm going to talk about transgenerational plasticities in snails, and also give you show you some preliminary results. Doesn't work. Sorry. Okay. It still don't work. Okay. Okay, nice. So sorry. So I'm going to introduce with the definition of phenotypic plasticity, which is the ability for a genome to produce multiple phenotypes in response to environmental variation. So it's also called within generation plasticity, as it occurs in one generation. And so in these kinds of graphs, one reaction norm corresponds to one genotype, where one environment corresponds to one phenotype, and for the same thing for the second environment. And it's very common across all the taxa. And it's been like 20 years that there were more and more studies about transgenerational plasticity, which was first studied through maternal effects. And it's when the phenotype of a generation can be influenced by the environment experienced by the previous generations. So it's also relatively common and in the majority of cases adaptive, but it can be also maladaptive. And so here we can see when the parental environment is different, the offspring reaction norm and genotype will produce different phenotypes. And so we can, there is some good examples. For example, in Viviparous lizard, where when the mother is experiencing predation from snakes, the offspring will have a different morphology. And in Arabidopsis thaliana, there is seasonal transgenerational plasticity in biomass produced. And one of the most interesting cases in humans are when transgenerational plasticity in nutrient absorption and retention. So for these studies, when grandchildren of great-grandfathers who experienced starvation during American Civil War the grandchildren are at a 20% increased risk of obesity and associated disease. And so all these studies are mainly conducted on 2 or 3 generations. So that raises many questions about the persistence of the plastic traits, their expression, and the evolutionary potential of such a transmission across the generations. And so this is an example of the few studies where transgenerational plasticity is studies over several generations. There is also one on nematodes, but this one is on aphids. And so one plastic trait in aphids is the wings. In a normal population, at a control rate, there is approximately 25% of winged aphids. And when you introduce the predator here in red, the rate goes by half the population who has wings. And when you stop the predation, it takes several generations to come back to a normal level. And so to explore these questions of transgenerational plasticity, we used Physa acuta, which is a small snail who produces a lot of eggs per week and has a short generation time. So it's very good for transgenerational large-scale experiments. Can be found everywhere in the world. Actually, my snails come from here in Montpellier. And the predation can be made either by fish or crayfish. We chose crayfishes, Faxonius limosus, which is an invasive species from USA. And we already know some plastic response in Physa acuta. For example, an increase in crawling out behavior when it's exposed to predation, which is the snail spend more times above the water. There is also a modification in shell morphology, size and thickness, and in life history traits. And what's very interesting from previous results, we also know plastic transgenerational responses for 2 or 3 generations. And usually it's the same traits that are plastic, but in the 2nd or 3rd generation, the intensity and the direction of the plastic response are totally in opposition to those within generation plasticity. So this was our experimental design. As you can see, there are 6 treatments. Actually, the original experimental design have very much more treatments. And so we have the P0 control line. And then for P1, P2, P3, the predator was added at the 1st generation, and then the 2nd, and then the 3rd. And there is also an accumulation line with exposure to predators constantly through the generation. And we also made separate lineage where the predator was removed after 6 generations of accumulation. And so we measured classic traits in plasticity, morphological traits, behavioral, and reproductive traits. And so these are the preliminary results. So here what we can see, we've got within generation plasticity, each time a predator was introduced in the different treatments. So here in the 1st generation, here in the 2nd, here in the 3rd, we've got some plasticity in the mass. So here it's a synthetic variable made from mass and length through an ICP, and then the individual means are corrected by the control line. So the means are relative to the 0 level here. But we can see there is within generation plasticity. So that was the proof that our experiment worked. There is a response. And what is very interesting, it's the increase in growth, so mass and length, following after like 2 or 3 generations. And the level of increase is really, really significant. So for the generation just after the exposition, the results are constant with our previous experiments. But here it's very interesting. And then it seems to go back to a classic level. And so we also added the accumulation line in dark blue, so always predation. And we can see the level is the same as when there was only 1 generation that was predated. And then when we removed the predator in the 7th generations, there was also a significant increase in the growth trait. And so the results are a little bit complicated with the other traits. Here is the reproduction trait, also a synthetic variable made with the egg number and the clutch number. And so I didn't show the accumulation curve because it's like nearly the same as the control. The snails here, maybe they were responded, the response was less intense. We can see maybe a pattern of ups and downs through the generation, but this may be also caused by natural variation. And it seems that there is not an impact of how, when the last generation was exposed to predation. So for the shell thickness, it's also the same kind of pattern, up and down through the generations, with some significant differences, and the accumulation curve being far above the others, but at a control level. So here the snails were not responding well also. And this is the different threat crawling out behavior. So 1 hour after predation exposure, we assessed the number of snails that were still out of the water to escape the predator. And so we can see that the accumulation line works very well, but there seems to be no difference among the treatments, whether the predation exposure was in 1st, 2nd, or 3rd generation, which is also quite weird because the P3 treatment was exposed last generation. Here it's the 4th. And so we expected like a more important proportion. So to discuss these preliminary results, we could see that growth traits can last for at least 4-5 generations. We need to do many analyses again, but it seems to be in line with our previous results for transgenerational plasticity in 2 generations. And maybe this is related to the refuge hypothesis in aquatic snails, where a bigger snail could be less likely to be predated. So it could be a response in the 3/4 generation. For the reproductive traits, there was no difference, but from unpublished results, we know that the transgenerational plasticity can be observed on 2 generations. We expected that the plastic traits could last longer, but it's not the case apparently. And for the different traits, it's really hard to draw a conclusion here because behavioral traits are known to be pretty labile, and also, even if there is transgenerational plasticity for behavior in the 2nd generation from unpublished results, we know that the rate of defensive behavior is very, very low. And for the shell thickness, so quite complicated to draw any conclusions. So it's important to be reminded that in situ, there is an interaction of both immediate exposure to environmental change and also the previous exposure from the previous generations. So it seems to be a question of energy allocation and maybe trade-offs between growth and reproduction. But actually, the main hypothesis for transgenerational plasticity is that it's in response to variable environments. So it works very good in heterogeneous environments. And some studies suggest. But it was the end. Just like different patterns of heterogeneity could produce different kinds of transgenerational plasticity. And thank you for your attention. And like I only presented like a very small part of all the study and all the, like even for this experiment, so I will be happy to answer some questions.
Thank you very much for the talk. I'm not sure I understood exactly how you calculated the plasticity, like the within generational plasticity. So did you measure like the reaction norm of each individual in terms of like this growth rate, like for each of the generation according to the treatment, and then you could compare this reaction norm to basically the control population? That's what's happening?
Yeah,
Okay.
I couldn't show it, but I did like the same kind of graphics, but for just one generation to compare the levels to the control. And when you look at these kinds of graphs, only one generation, you can tell, okay, here there is significantly more mass or that kind of, and it allows us to know if there is within generation plasticity, and for transgenerational plasticity, we need to look in the second or third generation.
And so related to this, could you check that actually, because it's related also to basically how the parents would react to their environment. So could you compare that actually the reaction norm are getting stronger for compared to the parents, or is it like more a population pattern that you observe here?
Oh, like actually for the generational pattern, it's the first time I did this. We just for the last 2 years we focused on 2 generations. But can you repeat the question?
So what I mean is that like the parents have some kind of plasticity to the environment stimulation. And I was wondering, like now you're trying to look at the effect on the offspring, like on the different generation. And I was wondering if you could actually link that to how the parents had their own response to the environment, basically. So that when you know, yeah, you know what I mean?
Yeah, I get it. Like, yeah, every generation can exhibit within generation plasticity, but also transgenerational plasticity through the last generations. And that's why here we just expose one generation to predation. Because if you start to like do something like the accumulation curve, there can be an interaction between immediate exposure and transgenerational exposure. There are some work on this. So it's very hard, even in experimental designs, to separate these 2 factors.
Yeah, I guess maybe in the one where you repeat the treatment, no?
Yeah.
Because here then every generation is stimulated, and you could see how the previous generation reacted to the same treatment. And I guess when you measure the traits also, you do it before and after exposure.
Yeah.
So you could also disentangle here what's happening before the stimulus, and it's only parental, kind of.
Yeah.
Or after the stimulus when they have the parents plus the exposure, the exposure behind.
Yeah, but I think this was also this was already made in Physa. But that's why I didn't show you everything. But we also got snail lineage with only 2 generations or 3 generations with accumulation to show the difference between 1, 2, or 3 generation of accumulation of predation exposure.
Yeah. Cool. Thank you. Really cool.
Welcome.
Okay, thank you for your presentation. I can imagine that it was a lot of work to do all the different generation. I just had a quick question because you did that on 7 generation maximum, but then how do you know that it's not, for example, short-term adaptation and that is really transgenerational plasticity? Like, did you look at the DNA or I don't know?
Well, I get your question, but it's kind of a broad answer. Like we also did a DNA analysis. So maybe I can show on the experiments. Like we took samples of these 3 lineages for 2 generations to do RNA-seq and ATAC-seq to see what genes or what chromatin regions are expressed, over or underexpressed. But I didn't get, I think, the end of your question.
it's like, for example, like your PACC plus treatment,
Yeah.
like at the really end, like how do you know that it's not maybe short-term adaptation, because after 7 generation it can already start to happen? So how, like, yeah, how do you know that it's transgenerational plasticity and not like the start of a really short-term adaptation?
Well, I didn't know it. That's really a good question.
Okay.
So I will think about this, but actually, I got backup from other studies to know the traits are plastic and are in response to predation. But yeah, absolutely, over all this generation of accumulation, it could be hard to tell. It's short adaptation. So I will think about this. Thank you.
Thank you.
Okay, so thanks again, Leo. So now we are going to listen to Aurora Garcia-Biero, who's from the Botanical Institute of Barcelona, and she's going to talk about genetics and painting lady butterfly.
Thank you. Good afternoon. Today I'm going to talk to you about the genetic architectures of migration in the painted lady butterfly. So migration is a complex trait. It encompasses a suite of morphological, behavioral, and physiological traits. And despite this complexity, migratory phenotypes have repeatedly evolved in distantly related taxa. So in order to better understand this, we can focus on the variation at the population level. In some species, populations have adapted to undergo different migratory directions, and thus resulting in the so-called migratory divides. Populations can also vary in the level of migratory intensity when they colonize new constant environments, such as tropical islands, which can even lead to the loss of migratory behavior. So differences in migratory traits can lead to temporal and spatial isolation. So what genetic architectures do we expect for these traits? Well, we can say that a trait is complex when it involves a series of genes or environmental factors. And so when complexity increases, we would expect to find an increasing number of contributing loci of smaller effects. And this is actually true for many complex traits, such as hate. However, there are some traits, some complex traits that have a mode of inheritance more similar to a simple inheritance of a single Mendelian locus, basically. And this is the case for low recombining regions, which are usually created by chromosomal inversions. These regions can span several genes, and these genes are transmitted together as a single unit. As an example here, we can see that there is a social polymorphism in ants, where the presence of a single queen or multiple queens in a colony is determined by a chromosomal inversion in chromosome 3. So the painted lady butterfly is a highly migratory species, and it offers a unique opportunity to look at the variation at the population level because of its nearly wide distribution. So like other insects, it has a multi-generational migration. So when the temperatures drop in the northern hemisphere, they travel south towards the equator and then back north again in the spring. But what happens with the migratory dynamics in the southern hemisphere? We know that migratory cues differ between hemispheres. So the magnetic field, the position of the sun, and seasonality in general is opposed in hemispheres. So we hypothesize that populations in the north and southern hemisphere have evolved the distinct migratory strategies in terms of orientation and timings. Painted lady butterfly also inhabits two tropical archipelagos in the Pacific, Indonesia and Hawaii. So in this study, we also tested whether they have independently lost the capacity to migrate. For that, we collected samples of all the interest populations, including some of the closest mainland populations of the islands, And we sequenced using RAD-seq technology this data set and whole genome sequencing the second data set. And also we made use of the already assembled Vanessa Cardi chromosome. Genome assembly. So moving to the results, we can see that principal component analysis clearly recovered 2 genetic clusters corresponding to North and South, and an addition of a few individuals in between those clusters. So it is in line with our hypothesis of the 2 independent migratory populations. We wanted to see where this differentiation lays in the genome, so we found this. This is a block of SNPs with very high differentiation, FST, that has these sharp edges and resembles the signal of a chromosomal inversion. This signal was also supported in this region by very low genetic diversity and an increase of divergence, especially at the edges of the inversion. Interestingly, those individuals located in between the 2 clusters were collected near the equator, especially in Ethiopia in the highlands, and they present higher heterozygosity values. So we suspect that these are individuals that are heterozygous for this inversion. We are currently characterizing the breakpoints of the inversion, and we think that one of them is intersecting this GABA-B receptor 2 gene, which is the receptor of the GABA neurotransmitter. And this neurotransmitter is involved in the neural network determining direction during flight. And this neural network has been extensively studied in the monarchs and other model species. So now we move to the Pacific, where this butterfly has colonized these 2 archipelagos, and we ask whether they're still migratory. And for that, we first had a look into the genetic diversity of the populations and the proportion of their genomes in runs of homozygosity. So we can see that Indonesia immediately comes as an outlier, which has almost half of the genetic diversity than the rest of the populations. And it also has up to 40% of their genome contained in homozygous tracks. So what this dark color mean is the proportion of the tracks that are shorter. So this excess of shorter homozygous tracks is usually associated with an ancient inbreeding, so maybe an ancient bottleneck. So it indicates that this population is likely being isolated for long periods of time. As for the Hawaii population, it has surprisingly similar levels of genetic diversity to the mainland Nearctic population. And it also does not show evidence for a recent colonization, because it would show up here as an excess of the longer tracks of homozygosity. And this is not the case. So we suspect that there is still some gene flow from the mainland population. And these results are also in line with the differentiation across the genome. So we can see up here that Hawaii is not so differentiated from mainland Nearctic, so from North America, whereas Indonesia has stronger differentiation with Palearctic. So these scans, what they suggest is that migratory laws may have a polygenic architecture, which is expected if you think that the colonization of a new environment not only involves the regression of migratory traits, but also adaptation to a new sedentary lifestyle. So at the molecular level, we can look at this adaptation as the selective sweeps. This is one of the ways adaptation can occur, where one variant reaches fixation really fast and drags with it other variants that are in proximity. And this is the signals that we can see in these plots, where in Indonesia, we have many selective sweeps, whereas in Hawaii, it seems that positive selection is less pervasive in this population, as we can see less selective sweeps. And most of them, the red ones, are shared with mainland population. So besides selection acting on different selective genetic features, there is still some shared similarity between the Go terms associated to these genes. And so it points towards a potential functional parallelism, but I'm still working on the statistics of this analysis. What we did have a look at is at the pleiotropy of these genes that are inside the sweeps, so the selected genes. And we measured pleiotropy as non-redundant GO-term clusters. So it is basically a proxy for the amplitude of the phenotypic effect that these genes have. And so we found that the genes inside the selective sweeps of Indonesia are far less, so significantly less pleiotropic than the rest of the genome. So in line with the theoretical expectations that we were mentioning before about lossy contributing with small effects. So basically the theory of cost of complexity. So to sum up, insect migratory species have a vast population numbers and panmixia. However, there is still some room for differentiation when migrating in the different hemispheres or when major geographical barriers prevent gene flow. We uncovered a non-megabase inversion differentiating the two hemispheres, pointing to a modular genomic architecture of this trait of sensorial and orientation adaptations, which is also the first migratory divide described in insects that we know of. Also, we discovered widespread genomic signatures associated with migratory loss, consistent with polygenic selection acting on small numerous effect variants. And last, well, disentangling genotype-phenotype associations is a very hard task, so hopefully this study will help doing so in the field of insect migration. With this, I would like to thank my supervisors and my colleagues, my lab mates, Gemma and Roger, who are actually here in this conference and have supported me a lot, as well as my collaborators in Edinburgh and in Uppsala, and the funding. And just for your attention.
You have some questions?
Thank you for the talk. Very interesting what you find with Hawaii and the mainland population. So I was wondering, is there some attempt to validate this prediction with going and monitoring maybe on the coastline some population to actually see if you can really dig and see that there is still mating happening between these two areas?
Yes, so I think that for the levels of genetic diversity to be as high as this is not required like a very constant input of newcomers, but just like every exceptional accidental dispersal would also help to increase diversity very quickly. So one way to test this would be, of course, to like go and the thing is, like, we should have some collaborators that live there year-round that could report increasing numbers of suddenly populations, painted ladies appearing in mass, which then would indicate that they just arrived with the winds. That would be the ideal. So far, what I'm doing is demographic modeling, which is far less interesting. But so I'm trying to model in a scenario where there is gene flow versus another one without. So working from the data first.
Yeah, that's cool. I was wondering, like, is there actually already some people who try this in the lab, just have it some kind of population and see how this index vary? Because yeah, I don't know anything in this index, so I don't know how strong, like, what does that mean when they vary?
The time that this has been analyzed is in the monarch butterfly, where they chose some of the in the Caribbean, there are some populations that are sedentary, and they try to place them into mainland again and see if they still retained the migratory capacity. And it's very cool, but we haven't done this yet. And I think that experimental research is really also the way to go here, to really test whether they're lost already completely the capacity or they still somehow retain the possibility to read the cues, like the photoperiod change and so on.
Thank you.
Hi, I think maybe I lost it, but do you have any idea, because you showed that there is one area in the genome where you have all these differences, do we know what is connected to these genes, or is there some job in the in the butterfly?
Yeah, I think there were around 330 something genes in that region, so it's quite wide. And we also did the Go-term association to look at the functionality of these genes, and they have a lot of them related with muscle development, fatty acid metabolism, and a lot of like external stimulus as well. And among these 300 genes, I still don't know like which ones are the most important ones in this case, but yeah, hopefully in the future we will continue with the inversion project.
No, but indeed, it looks like they could be connected to migration things, no? So it's quite interesting.
This is what we think. I think there is also a school of people in inversions that they think that inversions are always like very adaptive, but I think that also they may accumulate some mutations because they recombine very little. So part of the differentiation may not be adaptive inside the inversion. But I have the hope that we will find something interesting. Yeah.
Cool, cool. It was very interesting, eh? Thanks.
Thank you.
One last question. Nope.
Okay, so next talk is gonna be Emilio Egal from the INRAE. I wasn't sure actually. He's a PhD student there, and he's gonna talk about the Atlantic salmons and immigrants.
Hello, thank you for being here. So yes, we will talk about the fitness of immigrants, and we will see that it can depend on the population of origin in Atlantic salmon. But before getting into the heart of the subject, let me introduce you some concepts. So in heterogeneous environment, different population of a species may experiment different environmental conditions, leading to different selection pressures. And if gene flow is not too high, these pressures will lead to the evolution of locally advantageous traits and phenotypic optima for each of them. In the absence of other forces and constraints, this will result in a pattern where, on average, you will have a higher fitness for native individuals than immigrant individuals. This pattern and processes can define what we call local adaptation. So one selection pressure that can limit gene flow and influence local adaptation is the selection against these immigrants and subsequently their offspring. So the effects that immigration can have on recipient population depends on numerous factors, and these effects can be positive or negative. In positive, you can, for example, have an increase of genetic diversity, adaptive potential, and in negative, you can have, for example, the migration of locally maladaptive alleles, what we call migration load, and the deletion of local adaptive processes by submerging local adaptive alleles, what we call genetic swamping. Speaking of local adaptation, Atlantic salmon has been for decades a good model for studying it in natural systems. So this species is an anadromous species. So briefly, The life cycle will begin into the freshwater. So we will spend 1 to 3 years. And after, it can decide to go into the sea to spend 1 to several years for its growth. For example, here, you can see the blue area. And then it will come back to freshwater to reproduce. And what makes so special Atlantic salmon is its philopatry, known as reproductive homing, due to its capacity to find its river of origin even after traveling thousands of kilometers. But some recent studies showed that we have a percentage of individuals dispersing to new spawning grounds. So for this study, we will take advantage of these natural dispersal events to try to infer the proportion of disperser in a nivel population based at the southwest of France. Try to see if there is any bias in dispersal, for instance, linked to sex. After, we'll try to compare the fitness of natives versus immigrants. And within immigrants, try to see if there is an effect of the river of origin, as I said previously. And lastly, is there any differences in Bateman gradient between native and immigrants? But we will talk about it later. So for the sampling, as I said, we are at the southwest of France, just here at the Nivelle. For the adults, they had been sampled during 16 years at station capture here, Ochondoa and Ola. And for the juveniles, so the par, we had access to electrofishing sites, as you can see here. So with electricity, you can attract the juveniles, and these individuals has also been sampled during these 16 years. We also sampled 3 adjacent rivers concerning juveniles during the year 2021. After this, we extracted some fins and scale formed individuals. We extracted the ADN, and we genotyped some individuals with microsatellites, with a SNP chip, or for both methods, for certain individuals. So as I said, we have to identify the dispersers, but what is a disperser? What are the criteria who make an individual a disperser? Firstly, the individual doesn't have parents in the study river. For example, you can see the red individual and the yellow individual. You don't have parents in this river. For this, we used a method, a parentage analysis, to do this. So briefly, you have a cohort T, and you will try to assign these individuals at the cohort T-1. For example, one individual can have 2 assigned parents, 1 assigned parents, and 0 assigned parents. And as I said, if you don't have parents in the river, you are potentially a disperser. The second criteria, the different genetic makeup. So you don't see here the word, but it's different genetic makeup. Normally, for example, for this river, if you have a gray genetic makeup, these individuals should not match this makeup, and for example, belong to this river for the red individual, and for the yellow individual, the yellow genetic makeup for another river. For this, we used what we call genetic assignment test. So assignment test aim to assign an individual of unknown origin to its river of origin, based on its multilocus genotype and the expected probabilities of each multilocus genotype to belonging to one of the sources population. In our context, the adults are the individuals of unknown origin, and our juveniles of multiple rivers are our baseline. So for the first results, we found a total dispersal rate of 19.5%, and it goes from 4.2% to 10.2% for these 2 rivers. Concerning the dispersal, so when we are trying to classify the individual based on the years spent at sea and for the sex, we can see directly that we have a higher proportion of male in this category, so the smaller fish. And for the female, they are more on this category, the higher fish. And what we can see when we are seeing one category by one category, you can see that we have a sort of a malbiased dispersal, so a higher proportion of a disperser, for example, in the male rather than for the females. And this result is highly driven by the multi-single winter category, so the bigger fish, because you don't have a lot of multi-single winter males, but the proportion of disperser is higher here, for by here. So for the reproductive success, when we are trying to classify the individual just as native and immigrants, you can see directly, so you have the y-axis here for the number of offspring, that we have a reduction of fitness for the immigrant individuals. When we split this individual by finer scale categories, so for example, here you have the native, you have 4 categories of immigrants, you can see that you have an overall reduction in fitness, whether by males or females, but that this reduction is origin dependent. For example, the Nive River has a total reduction in fitness, whereas the Gave River didn't show any significant reduction compared to native individuals. Now let's talk about the Bateman gradient. Briefly, this Bateman gradient represents the slope between the offspring production and the number of partners associated. And this slope is in positive correlation with the strength of sexual selection. And as you can see directly here, that we have differences between the male here and the female. And then we try to compare the native immigrants for each of these sexes. And what you can see here directly is that we have an increase of offspring when we are adding a partner for the males, but that this increase is higher for the males. But the interesting result is for the male immigrants. For example, you can see here that we have a huge increase, so 83% for each additional mate for this male immigrant. So the take-home message, so for the first question, what is the proportion of disperser in individual population? We have seen that we have a substantial proportion of individual, 20%, and that this dispersal seems to be biased by one sex, so the male. So after, when we are trying to compare the fitness, we have seen that we have an overall native advantage, but that this advantage was highly dependent of the origin and other characteristics that I can't present here because of the time. And after, is there any differences in Bateman gradient between native and immigrants? We have seen that these slopes, so that this Bateman was stronger for immigrant male than for all other categories. Thank you for your attention. And if you have any question, let's go.
Well, it was very interesting. I have a question about the last result. So the Bateman gradient. First, it looks like that there is no really difference in the amount of partner that a native and immigrant can have, right? But for immigrants, individual increase more the number of offspring. That's correct. How do you interpret this? What does it tell us, this number?
For my interpretation, based on studies for me, so an immigrant has a disadvantage when he is going into a river that he don't know. But if this immigrant succeeds to reproduce with multiple partners, you can create different genetic combinations. So for example, if you are coming far from this river, you can have a sort of what we call heterosis. So you will increase your offspring, and this can be due, for example, to the bet hedging strategy. So you are taking risk to go into another river, but it can be like the casino, like a big prize if you are adding multiple partners for you.
So would it be correct to say that, well, most of the immigrants will have less offspring and mate with less female compared to the native, but the one that managed to reproduce many times, actually, they might bring some advantages to the female. Okay.
We'll have more offspring than for the local that mate with multiple partners because of this type of a combination, different combinations.
Very cool, thanks.
Thank you for the talk, that was great. Sorry, just formulating the question. You had immigrants from like local rivers. Could you tell if they came from farther away?
Yeah. Normally for our species, who is very known for its phylopatry, This is very rare because some studies show that you have a long distance migration, but normally for this species, it's around 100 kilometers. So normally we have the area. And the previous study showed that we have 5 metapopulations in France, and that's why we sample only this metapopulation in the southwest of France. But for the next year, I will try to compare all the metapopulations.
Thanks.
Question here and here.
Hi, thank you for the presentation. I have a question about phylopatry. And what do you think is the advantage of this strategy? Because to me, it seems that it comes with a clear cost in terms of inbreeding. So they're potentially more inbred than other, I don't know, less philanthropic strategies for migration. So what do you think?
I have the same question concerning the emerging. I can't study it here, but normally some studies say that as you know this river, it's better for you because you know all the risks. And you have two hypotheses, the pheromone hypothesis and the imprinting hypothesis. So when they are going down from the river to migrate into the sea, they are like chemical cues that can be recognizable when they are going back to this river. And normally, for this species, it's better to stay with your family. But for the problem of inbreeding, for me, that is a problem. And maybe that's why for the Bateman gradient, when you are doing multiple combinations, it can be better for you because you are mixing alleles, etc.
Thank you.
There is a question here.
Thank you for the presentation. The question is, you talk that the decrease of fitness was origin dependent.
Yeah.
And why?
Why?
Is there like human activities in some river that explain that?
Yeah, I think that it's origin dependent for some various reasons. First, I didn't present these results, but you have the sea age at maturity, you have the sex, etc. And for example, for some categories, you will have bigger fish. And if these fish migrate into my natal river, there won't be any problems for them because, for example, for females, you have a big fish, so normally the males will be attracted by you. So you have a multiple categories. And after, I think that we can compare the environment, for example, the granulometry, the type of rock, the type of flow, because, for example, if you have a huge flow, if you don't used to take this flow before, this will be a huge problem for you. And we have this type of case in some rivers in France. So you have a huge, strong local adaptation, and we don't have any migrants.
Yeah, it will be quick. So I was wondering if you had any data on this, I don't know, the offspring survival, maybe, or developmental speed, or growth rates. So basically, if the immigrants were doing as good as the local ones.
Yeah, I think that I can have access to this type of data, but I didn't do it for each river. But for the next paper, normally, I think that I will integrate this data to, for example, compare with the local adaptation, etc. But yeah, it will be very nice to do this.
Now we have 20 minutes of coffee break, and after for the other students on Tuesday to take the seats, it's over here that the Qu'est-ce que tout ça là? C'est pas mal, c'est fait comme.
Really sampling. Yeah, there's already something with the
Test, test, test. Test, test. Si on l'entend, c'est juste avec beaucoup baissé. Test. Test 1, 2. Test 1, 2. Ouais. Là, juste dans la salle, à droite, à côté des machines à café. Il y a une fontaine avec de l'eau froide. Test. Ah ouais, et tu vois au max, c'est fort. Arrête. Test. Test, test. Ça va. Hein. Je crois qu'on a un peu baissé le volume, mais vous entendez pas bien. Pour éviter que ça. Oui, normalement, je crois qu'on a baissé le volume.
Okay, so next, Ross Walker from the Heriot-Watt University in Edinburgh will talk to us about the role of pace of life in animal structures.
Thank you. Well, this is a lot of people. I'm Ross. I'm from Scotland, and I'm a first-year PhD student in applied mathematics at the universities of Edinburgh and Heriot-Watt. Today, I'm going to chat about my first project, which has been investigating how species' pace of life strategies may intrinsically influence their social network structures. Here's the thing. So as a brief background, pace of life history is a framework which broadly describes how species' life history trade-offs may collectively vary across this singular hypothetical axis. And this classifies species as relatively slow or fast depending upon the rate at which their life history events occur. And this framework is useful in explaining a lot of the sort of general diversity in evolutionary strategies that we observe in nature. Here, I'm going to talk about this specific sort of sub-framework, which is the fast-slow continuum. And this describes species as slow or fast, specifically on the trade-off between their recruitment rate and their lifespan. So a slow species is that which reproduces less frequently, but lives longer. And in nature, there's a huge amount of diversity across this axis, with some of the sort of extreme prototypical examples being Greenland sharks, which only reach sexual maturity after living for about 150 years. And then you have sand-burrowing mayflies, for which, after completing their molting stages, they live only for a matter of minutes, and they spend that whole time reproducing, which is quite an epic lifestyle. And so this diversity starts to motivate our main question, as in, could diversity in pace of life strategies help to explain the large diversity in social network structures which we see empirically? And these sorts of really broad conceptual questions are quite well answered using mathematical modeling. And so that's exactly what we do. I'm going to try and break down the different components of our model. The social structure itself, we model as a directed graph where nodes represent individuals and edges represent some kind of social bond. And the fact that edges are directed mean that we can represent a broader range of behaviors and structures, for example, dominance hierarchies, things like that. And then position along this fast-slow continuum, we model using a continuous time Markov chain. And this utilizes a single parameter C to essentially equally scale the birth and death rates of the population. So a higher value of this parameter C corresponds to higher birth rate, higher death rate, which is a faster pace of life under this fast-slow continuum framework. And this particular model gives us stochastic dynamics, which also keep the population size as an integer, which is useful for integrating with the social structure. And so when an individual is chosen to die, we influence the social structure by just straightforwardly removing that node and all of its associated edges. But when individuals are born, we need to consider the notion of social inheritance, which sort of describes if or how social bonds are passed from parent to offspring. So here, for the sake of time, I can just talk about the two extreme scenarios we have, which we call full and zero inheritance. And this somewhat straightforwardly just represents the cases where offspring are assigned all or none of their parents' connections at birth. So collectively, these three components allow us to run demographic processes on a social network for various pace of life. But social structures also vary due to social dynamics or social behavior. And so we also want to include that within the model. And for this, we utilize the social organization model of Pfefferman and New 2007, which I've got a citation for at the end. And in this framework, they assign individuals with a sort of social preference, which gives them a way of ranking the quality of their connections. And so, for example, here with popularity, higher scoring connections are those which are, well, more popular. They have a higher in degree within the network. And for closeness, it is individuals which are more integrated within the social network who are more valuable. And this may represent individuals who, say, have better access to social information on, say, foraging or predator locations. And so equipped with these preferences, individuals, while aiming to keep a fixed number of connections, will remove their worst scoring connections and then pick new random ones throughout the network. And they will iterate this process periodically to try and sort of optimize their local networks. And this is a sort of form of individual behavior influencing group structures. So these are the main aspects of the model. Essentially what I have is a dynamic social network for which we can vary pace of life, social inheritance mechanisms, and social preferences. And we assume, for simplicity of analysis, that each group that we consider is sort of homogeneous. So they have the same pace of life, same inheritance strategy, and same social preference. So to take you through some of our results, we find that when there's no demography, the social networks can effectively self-organize using these social preferences. So in the case of popularity preferences, which are shown here, we find a really centralized structure emerges, where everyone is connected to these super popular individuals, and everyone's social preference is sort of well fulfilled. Then as we increase the pace of life or add some demography, this centralized structure starts to decay. And this is because at faster pace of life, there's a higher rate of deaths. And so these popular individuals tend to die off more frequently. And when this happens, the networks have to sort of reconverge towards the centralized state on the left. Yes. And this takes time. But with faster pace of life, this sort of resetting of the convergence happens more frequently. And if you look at this axis below, you'll see that this breakdown in the structure actually happens quite rapidly. And so to quantify this effect, we use measures from social network analysis. So that rapid decay from the previous slide is quantified in that top left panel there, which shows the centralization of the network decaying somewhat like a power law. And if you also notice here that with full social inheritance, the centralization is consistently a little bit higher. And this is because offspring are now born with, well, the connections of their parent, which have already been somewhat optimized by their parent. And therefore, each individual starts off in a better position in life, and the convergence of the network is consistently a little bit further along. Also notice that in these faster systems where there's a lower level of centralization, there's greater cohesiveness, which means on average, the distance between individuals in the network is smaller. And this could mean that things like social or epidemiological contagion could spread more effectively throughout these networks. Then when we have closeness preferences as well, I know there's a lot of results, so if you're lost, just ask a question after. We don't see it as extreme of a pattern, as you can see by the boring curves on the right. But we do notice this interesting phenomenon that the social inheritance mechanism can actually reverse the direction of effect of pace of life on the networks. So if you look at that top right panel, there's a slight increase in the centralization, a slight for full inheritance, and a slight decrease for zero inheritance. And so essentially what I'm trying to get to is that social inheritance mechanisms, social preferences, and pace of life all interact in quite complex ways. But if I'm to summarize it in some sort of concise way and give you one thing to take away from this talk, it would be this, that for slow species, their network properties are determined primarily by social preferences, but for fast species, it is social inheritance mechanisms. And this is sort of verified in the full paper, where we consider additional preferences and additional inheritance types. And this can have some evolutionary implications as well. Since social network structures influence fitness at multiple scales for a wide range of social species, we find that, or I have 3 minutes left, really? Wow. I definitely missed something. Essentially, we may see selection for particular properties of the social network. And what our results tell us is that the selection on slow species may be realized on their preferential behavior, but for fast species, it may be on their social inheritance strategies, since these were the mechanisms that were able to more directly influence the network structure. And therefore, hiding that, pace of life strategies may influence selection on social behavior. In the other direction, social behavior may influence pace of life. Because, for example, with popularity preferences, we could only see highly centralized structures at a really slow pace of life. And what this implies is that, well, if there's an evolutionary advantage to having a centralized structure or being connected to really central individuals, then there may be some selection for a slower pace of life based on your social behavior. And this is because the networks can more effectively self-organize. And so collectively, what this implies is that social behavior and pace of life may co-evolve. And this work is sort of able to quantify this in some sense. So I've gone through that way quicker than I expected, which is unexpected for these sorts of talks. So importantly, I want to say thanks to these 4 faces at the top. This is Andy, Matt, Xander, and Nina. They've been fantastic while I've been working on this, especially as a mathematician getting into ecology, it's sometimes a bit scary. There's a link to the preprint if anyone's interested in other results we have. Also, if you want to just chat to me about it, then come see me during the opening cocktail later, which I'm desperately looking forward to right now. All right, thank you.
Hi, it's very cool, everything. I have just one comment. So for social inheritance to happen, you would imagine that you need, what do you call it, the long and slow species? Because the way it works, like maybe you live with your mother, she doesn't die before you, so there is overlap in generation because she needs to transmit the social contact or support or friends, she needs to teach you to be socially transmitted. So somehow, if you want slower species might be more, might be more social inheritance happening. What do you think?
Yes, I mean, essentially the way we model it is it's like independent things, right? Like in reality, there's quite a strong correlation between slower pace of life and like parental care and parental inheritance. I guess what we're able to, like, this is all sort of seeing the conceptual implications of what it means to have more or less social inheritance, right? And some, like, this model is like super modular as well. And so what we're doing is we're trying to expand it in different ways. And it's looking at, for example, when there are stronger parental bonds for slower species, things like that. And in the main paper, we also consider the case where there's like a social bond which is actually given to your parent as well, which may be a bit more of a behavior which is consistent across the whole fast-slow continuum. And that is something that I guess I'm going to make you read the preprint to hear about, because there's quite a lot to get into that.
Thank you.
Good catch.
Yay. Thank you for the talk. I just had, just out of curiosity, if the populations is not, it's literally in the middle, meaning it's neither that fast nor that slow.
Yeah.
And the span that you're saying, what do they do? Do they do both or don't do anything?
Well, yeah, so essentially, I maybe didn't explain this part very well, which is why I was so quick. So, like, really slow species, you basically just need to look at their social preferences. Really fast species, it's just their social inheritance mechanisms. And the fast-slow continuum sort of gives you, like, how relatively important are these features. So there'd be a more of an equal split between these two features for sort of intermediate species. But like, it's not entirely straightforward in the sense that some social preferences will be a bit more like influential to the network structure than others. So for example, in some papers, they found that when networks sort of organize according to closeness, there's not really much that can actually change in the network because of the way that closeness works is that it's a very sort of fragile metric. And that when individuals try to attach to the most close individuals, they change how close those individuals are, and there's a whole bit of messiness there. And so that would be an example of a bad social preference. Whereas popularity is a good social preference because it can create really strong structures, such as the centralized one. And then social inheritance patterns as well. Some of them, like the parental one, for example, doesn't have much of an influence for some of the results. I was actually really lazy and didn't even remove the parental inheritance curve from these plots. So you might see in that bottom right curve, there's like a red one just under the yellow one, which represents the parental case, but you wouldn't see it, so I didn't remove it. So yeah, hopefully that blathering has somewhat answered your question.
Thank you.
That was a really interesting talk. I was wondering if you know how this kind of slow, fast continuum would, and like social preferences would work in like eusocial species like Hymenoptera. So like species where there's also like asymmetrical relatedness things going on. Was that too ecology?
That's very ecology.
Sorry.
As in, there's like, so for example, like with eusocial insects, there's like really sort of rigid social roles. Is that like in dumb terms exactly what you're saying?
Yes.
Okay, cool. So that goes back to what I'm talking about with this model being super modular, right? And in the paper, I also try to say that like, this is really general. It could be applied to specific species in this way. So for example, you could even add social preferences or this new component, which is saying that they may want to associate their social bonds to those which are nearby them spatially or something like that. And so we could represent those sorts of things in the model, and that could give some sort of new interesting theory as well. As to what I think it would actually do, it depends. Like, for example, if you're forcing following based like sort of, or some, you need to enforce some sort of like, what am I going to say? Yeah, I don't know. It's a bit difficult. But thanks for the question. That's some cool things to think about.
Hell yeah.
I was wondering if you've looked at the variation between male and females. So like in slow species, like primates, there's a lot of variation in social networks of the males and females.
Yeah, I've not looked at that, unfortunately. I mean, I've looked at something which is probably completely different, which is like when species have like a 50-50 split of different social preferences, which is somewhat getting at the idea of having a sort of dyadic split in behavior. And so far, I've not seen anything particularly unintuitive. You get something that would be like, if it's half popularity, half closeness, then you get the curves that are like halfway between the different preferences sort of thing. There's not any particular non-linearities that I've noticed. But again, I go back to the modularity, where if there's any like specific social niches for a specific species, then that could be fairly easily represented and studied here. Cool.
Thank you.
Thank you very much.
Okay, so next is Lina Ben-Lemli, and she's going to talk about parasitoid and parasitisms in aphids. And she's from the I don't know how to say it in English, CBGP, in Montpellier also.
Thank you. So yeah, my name is Lina Ben-Lemli, and I'm going to talk about the effect of parasitoid venom on the success of parasitism of aphids protected by the symbiont Hamiltonella defensa. This is my second year of master's internship that I'm doing here in Montpellier in the Center of Biology and Management of Population with my supervisors, Maximilien Adam and Henrik Frago. First of all, I want to talk a little bit about predators, parasitoids, and aphids. Aphids are herbivorous insects and a major crop pests. Their population is controlled by predators like the ladybugs that you can see here, but also parasites like the parasitoid wasps. These natural enemies are used in biological control to reduce pest population in agriculture. I want to zoom a little bit on aphid parasitoid wasps. So during oviposition, they inject an egg and venom into the aphid. The venom then modifies the host's physiology, which allows the development of the parasitoid larva within the aphid. The parasitoid larva then feeds on the aphid from the inside until its death. So the aphid then becomes a golden mummy that you can see right here. And then at the end of this cycle, an adult parasitoid emerges from this mummy. However, some aphids possess the symbiotic bacterium Hamiltonella defensa, which protects them against parasites. This symbiont produces toxins that kill the parasitoid larva. This protection have been studied for more than 20 years, but there is still no study on the correlation between parasitoid venom and the presence of Hamiltonella defensa, even though venom is really important for parasitism success. We can highlight that there is a high specificity between Hamiltonella defensa, so the symbiont strain, and parasitoid genotype. With one genotype of parasitoid, we can have different level of success of parasitism depending on the strain of the symbiont that's in the aphid. For example, with this specific genotype of parasitoid wasp, we can highlight that this genotype is able to successfully parasitize aphid possessing the Hamiltonella defensa strain A in green. However, it's not the case with the strain B in orange. So the success of parasitism depends not only on the presence of this symbiotic bacterium, but also on the strain. Denise and her collaborator wrote a paper in 2017 about parasitoid gene expression that changes after adaptation to symbiont protected hosts. So they were rearing parasitoid wasps on aphid possessing different strain of Hamiltonella defensa. This was an experimental evolution. You can see the colors right there. So parasitoids that were reared on aphid with no symbionts are in black, and parasitoids that were reared on aphids with different strain of the symbionts are in pink and yellow. So you can see on this graph the percentage of aphid mummified, which means parasitism success, depending on the aphid hosts. So at the beginning of the experiment, we can see that parasitoids could parasitize a lot of aphid with no symbiont in black, but there were only a few mummies among the aphid possessing the symbiont. After 10 generations of experimental evolution, parasitoids showed increased abilities to parasitize aphid possessing the strain they evolved with, which means that if parasitoids evolved with the pink strain, they increased their ability to parasitize aphid possessing the pink strain. And the same goes for the yellow strain. They also showed that parasitoids adapted to protected hosts showed an overrepresentation of venom-related genes compared to non-adapted population, which means that this adaptation may be linked to the venom. And that leads us to our study hypothesis, which is that parasitoid wasp venom interacts with Hamiltonella defensa. We are wondering if Hamiltonella defensa protects aphid from parasitoid wasp venom. And we are also wondering if the protective effect of this symbiont is strain specific. For the materials and methods, our living organisms, the aphid and parasitoid, come from Professor Christopher Berger in Switzerland. We have been rearing Lysiphlebus fabarum as a parasitoid that's able to parasitize the aphid Aphis fabae. And we have 3 different lines of aphids. The first one with no symbiont and 2 lines with different strain of the symbiont. These lines are clonal lines. They are genetically identical and they differ only from the presence and the strain of Hamiltonella defensa. So this specific parasitoid is able to parasitize aphids with no symbiont. It's also able to parasitize aphids with the strain A of Hamiltonella defensa. However, the success of parasitism of aphids possessing the strain B is really low. You can see the results from Caetano and Warburger from 2015 here. It shows that parasitism success is drastically reduced in the presence of strain B in orange. So for the methods, we start with a dissection of the venom glands of the parasitoid wasp to collect the venom glands. And then we inject in the aphid either PBS, a saline solution, which is the control, or the venom solution. I injected more than 600 aphids and measured mortality 3 days after. Here is a video of the micro injection. You can see a micro capillary glass going inside the aphid, and then it's filled with PBS. I can assure you the aphid is sleeping, it's not feeling anything. So we have 6 treatments with 2 injections. We have the PBS injection, that's the control, and the venom injection, and 3 different lines of aphids. The first one with no symbiont, the least protective strain of Hamiltonella defensa, and the last line of aphids possessing the most protective strain of Hamiltonella defensa against our parasitoids. For the results, here you can see on the y-axis aphid mortality as a percentage according to the injection type and the aphid line. On the x-axis, we can highlight that for the line of aphid with no symbiont, there was a significantly higher mortality with venom injection. And we have almost the same result with the least protective strain of Hamiltonella defensa. There is a higher mortality with venom injection. But then, if we look at the most protective strain, the symbiont B, venom has no effect on aphid mortality. So maybe aphids are immune against venom in the presence of this symbiotic bacterium of this strain. If you remember results from Caetano and Warburger in 2015, they showed that parasitism success was drastically reduced in the presence of this strain. And we just showed that venom injection has no effect on aphid mortality in the presence of this strain. So maybe venom is a target of symbiont conferred resistance. We can conclude that there is indeed an interaction between Hamiltonella defensa strain and the parasitoid venom. There is also a conclusion that we can say about the protective effect of Hamiltonella defensa that is strain specific and can counteract venom activity. For the next steps, we are going to investigate the effect of parasitoid venom on the concentration of Hamiltonella defensa in aphids. And we are trying to demonstrate that venom enhances parasitism success against aphid protected by this symbiotic bacterium. I would like to thank all the people that helped me during this experiment, especially my mentor, Maximilien Adam, who's just here. And thank you all for listening.
can.
Thank you. Thank you for the talk. That was really interesting. I have like two questions. First of all, if symbiont B is kind of successful, why is there symbiont A? And secondly, do the parasitoids just increase the venom, or is it the same amount but it's more toxic?
Can you repeat the first question?
So symbiont B is resistant to the venom, right? Did I get it correctly?
Aphid possessing this
Yeah.
strain, yeah.
Yeah. Then why is there's just that kind of symbiont? Is that is it just natural variation that there are two types?
Yeah, in nature, you can find aphid with without Hamiltonella defensa because it's a facultative symbiotic bacterium. And in the populations that you can find that have Hamiltonella defensa, they are different strains.
Okay, so natural variation.
Yeah.
Okay, cool. And like the efficient venom that kills them no matter what, is that just more toxic or?
I think what you mean is that are some venom more efficient than others?
So there was a part that the venom was overrepresented in the
Yeah.
in the parasitoids that deal with protected aphids, right? So if the venom is overrepresented in them, does that mean that they what does that lead to them producing the venom?
They just showed that there was an overrepresentation of venom-related genes.
Okay.
So it doesn't, I don't think that it means that there is more venom. It just means that the genes are overrepresented.
Okay, so it's not known. It's overrepresented, but we don't know what that overrepresentation does to the venom. Is that?
So there was an overrepresentation of genes, and they are saying that these genes are related to the venom, but we still don't know about the quantity of venom.
Okay, thank you.
Is that clear?
Thank you for your presentation. I just have a question. Maybe you explain it. Do you know how the bacteria actually can protect the aphid? What mechanism can explain these levels of protection?
So the bacterium produces toxin. For the bacterium to be able to produce this toxin, it needs to be infected by a virus. And when it's the case, then the toxins kill the parasitoid larva.
On this slide, if I interpret it correctly, so it also shows that when you select the parasitoid for a given strain, then they lose the ability of infecting aphids that don't have the symbiote, which suggests that there's a huge cost to kind of for this adaptation if then you end up with another strain. So do you think that this explains why we have this polymorphism in nature, is that you can tackle one, but then it's so specialized that it's no longer working with all the other, not without the endosymbiote or with another symbiote. And I mean, did you look at this in natural population to try to understand how this thing is evolving?
No, I didn't. But I think that Professor Christopher Berger is doing experiments on different populations in different countries. So maybe we will learn more about this. But it's really interesting that you highlighted that. Thank you.
Okay, so finally we are gonna listen to Pierre-Alexandre Quittet, who's a PhD student here in the CEF, and he's gonna talk about a cute little mammal, the alpine marmot.
Thank you, Lisa. So this is my first time with the microphone, so if I speak too loudly, please tell me. So I feel super happy to have the opportunity to present you the results of my first year PhD here. Thank you, thank you. And so today I'm going to talk to you about the evolution of body size in response to climate change in the alpine marmots. So body size, okay, obviously. Sorry for that. And this should work. Okay, so body size has been proposed as the third universal response to climate change, alongside change in geographic distribution of the species and an advanced phenomenology of the biological calendar, all right? So presented like that, all of these responses are implicitly considered adaptive, so it would be beneficial, for example, in that reduction of body size to be smaller, right? But that's kind of a mismatch of what we know about body size, right? Especially in mammals, when usually bigger is fitter, and with body size being associated positively with fitness, okay? So although it has been observed in many taxa, this reduction of body size is still unclear if it's always adaptive and which are the mechanisms underlying those changes, okay? So to be a bit more specific, we would expect body size reduction to be adaptive if natural selection favor that body size reduction. So we would expect higher fitness for smaller individuals compared to bigger individuals. And if it were non-adaptive, most likely due to environmental stress, for example, we would expect bigger individuals to have higher fitness than the smaller ones, okay? And considering the mechanism of changes, super simplified, but I would distinguish between plasticity, so changes in body size and the phenotype without genetic changes, and evolution, so implying genetic changes, okay? So, as I said in the introduction, body size is super strongly linked to fitness in mammals, and it's even more true in hibernating mammals that needs to accumulating fat in order to bear the cost of reproduction. Okay, so this is why I work on the alpine marmot, and so it's a socially monogamous cooperative breeder with the social unit being organized around one dominant couple that monopolize reproduction through aggressive behaviors. Okay, subordinates adults that does not reproduce, and the juveniles. So what makes the marmots a good model also for this question is that it's strongly impacted by climate change, with change in the social structure, with group size tending to be smaller and smaller with time, and a variation in fitness also, notably the survival of the juveniles. Okay? So to summarize, my question was, is there a role of adaptive evolution, so driven by natural selection, in the response of body mass to in the response to climate change? Okay, so I decompose this into three sub-questions. First, do we observe the size reduction? If so, is this adaptive or not, depending on which phenotypes is favored? And do we observe evolution? Okay, so genetic changes toward individuals smaller or bigger. So how do we do that? I use a quantitative genetics framework. So basically the idea is to decompose the phenotypic variance of a population. So in other terms, it means answer the question, why do individuals differ in their phenotypes, in their characteristics, okay? So part of this difference can be due to genetic differences between them. So what we call additive genetic variance. So this is the quantity that we're the most interested in, because this is the one that selection will act upon and create genetic changes, okay? And this is super closely linked to the concept of breeding values, which represent the effects of one individual's genotypes on its phenotypes, okay, characteristics. So, okay, and then individual can differ because of any other things, right? So environmental, what we call environmental variance, so it could be maternal effects, food availability, early experience, anything. But we're not that interested in that quantity because it's less straightforward. It's linked to evolution or genetic changes. So we use an animal model. It's a kind of mixed model that, with the help of phenotypic data and pedigree, a genealogy, allow this variance partitioning that I just showed you, the estimation of selection and of breeding values, this genetic contribution of the genotypes. Okay. So for the phenotypic data, I worked on a 34 years monitoring of a population of alpine marmots. For this study, I only focused on juveniles. Okay? And for this presentation, I only focus on the body mass as a trait, okay? Which is a proxy for the body size also. And for the pedigree, we know basically all of the individuals of the population, so we are able to reconstruct the relatedness between each pair of the member of that population. Okay, so we'll not dive into how does it work, but you just have to believe me at this point. But now we can infer some interesting genetic quantity with that. Okay, so now the interesting part, the results. So we did observe a decrease of body mass with time of the juveniles, and more interestingly, although selection favor heavier individuals. So it's a bit counterintuitive. It means that although body mass is decreasing, this is still the heavier individuals that reproduce or survive the most. Okay, so we have a non-adaptive decrease in body mass. That's what we showed there. And right now we wonder if genetic changes have anything to do with these changes. Okay, so first we need to know if at least part of the variation of the body mass in the marmots are due to genetic differences. Okay, otherwise it won't be transmitted to the next generation. Super simplified, but that's the idea. So first, well, so we did that various decomposition that I showed you, and I don't know how to point. Okay, this is the one that is important. This is our additive genetic variance due to genetic differences between individuals. So it's about, we say that body mass is heritable, and it's about 0.3, which means that 30% of the variation of body mass in our population is due to genetic differences between our marmots. Okay, so it means that body mass can potentially respond to selection and trigger genetic changes. Okay, this is the next step. So we estimated with the animal model the breeding values of our population across time. And what we showed is that breeding values, a genetic contribution to body mass, tends to participate to the traits more and more positively, which means that across generation, this is the alleles that favor a heavier mass that are favored across generation. Okay, so we know the term. This is an evolution towards heavier individuals. Okay. So to summarize the results, we observed a non-adaptive decrease of body mass, partially buffered by selection, by natural selection and by evolution. Okay, and I didn't say that, but we showed that it was not drift. It's likely due to selection. But I can okay. So the discussion. So this kind of pattern, evolutionary trajectories, where is called cryptic evolution, when genetic changes goes on the opposite direction of the phenotypic trends. So where environmental effects mask the genetic changes under the population. Okay? And it's kind of a cool pattern because we have in ecology paper where we can call adaptive storytelling, for example, where in the discussion or anything else, they can talk about adaptiveness or evolution without explicitly measured neither selection or neither genetic changes, and only based on phenotype. So this is a pattern, evolutionary pattern, that highlight the cautiousness of on not inferring anything genetics based only on phenotypic data. Okay. And so now the question is, why do we observe this body size reduction while evolution is pushing toward heavier individuals? Okay, why that? So based on the literature, we know that climate change is reducing snow cover during winter, and it's a bit counterintuitive, but because there is less snow, it's colder in the burrows because snow for isolation. So the less snow, the colder the burrow. And more on that, I said that in the introduction, but there is also reduced group size in the marmot, so it's less potentiality for social thermoregulation. So individuals keeping themselves warm, basically. Okay. So all of that will create an increase in energy expenditure to maintain a viable body temperature during hibernation. So marmots will emerge from hibernation in poor body condition, and because they reproduce super shortly, a few weeks after emerging from hibernation, said that too much, females or mothers will, in poorer condition, will produce lighter offspring. And our contribution to this is that we show that part of this deterioration, environmental deterioration effect on body mass is partially buffered by genetic changes that goes in the opposite direction of those environmental effects. And that's it. Thank you very much to the organizers that did an amazing job. Thank you for the field worker also. And I forgot, but Christophe and Céline also, that are my supervisors and that are absolutely amazing. They're not there today, but thank you to them also. If you have any question, I'd be more than happy to answer them. Thank you. Yeah, sorry.
Hi. Thank you for this great talk. It was really great. I just had one question. I think I asked you this before, but I forgot the answer. It's about the graph on the right, the breeding values. There is kind of an N shape.
Yeah.
Why is that?
Yeah, but I simplified the story for the purpose of this talk. But this is currently what we're trying to identify, and I'm currently because so everyone can see it. But we have a strong response to selection till 2005, maybe a bit more, and then it can at least slow down. Otherwise, it get super flat. Okay, and so I'm currently in a we don't know yet, but that's the enigma we're trying that's the enigma we're trying to resolve right now. So it might be, in my opinion, that those social structure differences, because litter size are decreasing, group size are decreasing, the number of helpers, so non-reproductive adults that helps for the pups, the juvenile survival, all of that are decreasing, and then it's kind of stabilizing. So right now, and we have preliminary results for that, but selection might act super differently depending on the social structure. So it might be because of that, because our hypothesis, it is a big story, right? I need to test it formally, and that's what I'm trying to do. But maybe we reach a point where we're kind of at the equilibrium, and because of the social changes, we don't need no more evolution, because we're in smaller group. Maybe it's less interesting to be, that's kind of story. So, but I can't tell more than that. But it's probably because of social changes, because our preliminary results show that it has a tremendous impact on selection. So probably on response to selection. Then that's the idea. Sorry, so long this response. It was clear. Yeah.
So that's gonna be on the same graph.
Yeah.
Because that looks like that's a super interesting point that you just made, but that looks to me like a nonlinear relationship with that kind of shape.
Sure.
Could it be simply that the weather condition in those years was just better or worse, and that had a direct impact rather than being an evolutionary thing?
Yeah, totally. But what we showed is that if you plot the snow cover, we know that the snow cover is a main abiotic environmental deterioration that affect the marmots. Okay, and it's a linear trend that goes like this. So it's still decreasing at basically the same path, more or less. Okay, so it might explain part of this variation, but I don't think it explains all of it. If that was a question, right? But it's definitely, variation because of the abiotic environment. But it does not explain everything for that now.
Thank you.
Because of those trends, right? That doesn't look like that at all. Like. It's fun to see.
Thank you for the presentation. You want to ask? So it's interesting because in your introduction, you said that basically when we look at this decomposition of variants, we are mainly when we study evolution, we are mainly interested in this genetic part of the equation because the rest is not so important or is not that strong. And at the end here in your result, we see that this rest of the variation is actually 75% of the variation that is not explained, and that the genetic in a certain aspect explain very little. So I was wondering why not consider, and then you have the sentence saying that basically here evolution does not buffer the decrease of the body mass.
It does buffer, but not completely.
Yeah, yeah, exactly. It does not completely buffer. But why not seeing also this environmental part of what evolution can act on? Because there is this environmental inheritance, and basically, yeah, the marmots who are born somewhere, maybe they get some kind of specific type of snow, and they are like, in a way, they could also decide to disperse and go in places where there is like better snow cover or something.
Not really.
If they are able to
okay.
see it or whatever, but I don't know. I think when studying evolution, sometimes we focus a bit too much on this genetic aspect, and the environmental is also part of this evolution. So maybe evolution, like not like in that sense that it's not buffered by evolution, but maybe it's just that evolution is not selecting just smaller body mass. You know what I mean?
No, I don't think. Well, I followed you until the end of the. Why would?
No,
why
so
would,
it's
why,
because it's just a bit of like.
Why selection would favor? So I don't.
What I mean
know
is that
on
the full
what you
process of evolution is this decreasing slope, and it's not necessarily this positive slope of the scale mile of like the positive slope of the heavier individual will produce more.
Yeah.
That's
I'm so sorry. I have trouble understanding.
It's just provoking thoughts. It's
But I do agree with you with the genetic part that maybe has too much focus on that. Actually, my thesis is about indirect genetic effects and how social environment will affect evolutionary trajectories, everything on that. Okay, but okay, but I'm not sure to see your point in the end, but it's not because it's decreasing that it has anything to do with the relation between fitness and the traits,
Yes,
right?
yes,
You know
yes.
what I mean? It's just
Yeah,
environmental stress.
yeah.
And for the mammoths, it's kind of convenient because they don't move. Basically, they just inherited their burrows, and all of that variance is captured in the maternal effects. That we put in that model, and it just it's both the maternal effects, the common environment effects, the territory effects, everything on that. So part of what you said is inside
the environment.
Inside this. It's probably why it's that big, actually. But now the next part also is to decompose this into the genetic components, non-genetic components. It would be pretty interesting. But yeah, I hope I at least. But we can discuss after that if you want.
Completely. Cool. Thanks.
Thank you for the question.
I was wondering, did you try to look differently at selection on body mass, on selection on relative body mass?
On relative body mass?
Because if it's all about competition for food when they just come out from hibernation, maybe what matters is not to be big, but just bigger than the others.
Yeah, sure.
So I was wondering, like, because how does body mass get selected? Like, again, I think, like Marta said before, it's probably all about food. So what's actually selected is probably not the growth as such, but it's probably like eating quickly or eating different kind of vegetation that they would usually not do, or something. I know nothing about mammoths, but I thought they were eating chocolates. But
I that somewhere.
But yeah, so like, did you compare to try to get a bit to the bottom of the proximal, like the ultimate trait?
Yeah, sure. And it doesn't change anything. We have basically the same evolutionary trajectories with a body mass scaled per cohort, per year of birth. So your body mass relatively to the mass of the other of your own year of birth. And it's the same pattern, okay? It's exactly the same pattern. We have the same breakpoint. So we didn't see much there, but at the almost 2005.
And is it that the relative body mass is really correlated to the absolute body mass in that particular case?
Probably, yeah, probably. Okay. Yeah, I didn't explore that, but that's probably the case, right? I can't see any other reasons, but thank you.
Yeah. Thank you, Pierre, again.
Thank you.
Thanks a lot for listening to all the talks. And now it's really time for a small welcome.